Hi everybody, welcome to our second podcast on macromolecules. In this one we're going to focus on carbohydrates and lipids, but again remembering that this is all about the matter, right? So again, got to bring back the Harold's cat. So let's get grooving. So here we are with proteins. Let's jump right in, again remembering those 4 factors from the last podcast that we want to focus on. Okay, so what they're made of, their functional groups, the monomers that make them up, what their overall functions are, and how they work. So again, the general info for our proteins. They are hugely complex. They're probably our most complex one given the amount of differences we can have in them. We have carbon, hydrogen, oxygen, nitrogen are the main elements found, but we do find sulfur too in 2 of the amino acids. And they are, I don't know, I don't even think I can understatement this enough or even overstate it. They're huge. They're important. They do everything, and they are a big, big deal. So they're monomers. They consist of amino acids joined together by that very specific covalent bond called peptide bonds. Okay, so if we have one amino acid over here, we hook it to another amino acid, again, dehydration synthesis. We're going to do a condensation reaction. We're going to pull out a water molecule, and we are going to combine the peptide bond. Here is the carbon that came from the carboxyl group of 1 with the nitrogen of the amino group on the other, and boom, right here, super duper important. And that creates what we call a dipeptide because we have 2 amino acids linked together. 3 tri, okay, and so on. Okay, so amino acids are kind of a big deal, okay? 20 of them exist. Some are then converted. So there is, in essence, I guess technically 21. Some people could even argue 22. These are not naturally occurring. The body actually will take an original amino acid and convert it over. So they all contain that amino group, that carboxyl group, to a central carbon atom, and then you have that R group, that varied R group that exists. Okay, so here we have some of the amino acids, and they all have very, very distinctive properties, from some being acidic, some being basic, some being positive areas, some negative areas. But regardless, you can see those commonalities that exist. Okay, so we have polar regions. We have some unpolar regions. Here are some others. There's one of the ones that contain sulfur. Cysteine is one of the ones that contain sulfur. Okay, and we have some, notice pHs are a little varied with them. Methionine, there's your other sulfur right there. Okay, so methionine, that carries a very important role in protein synthesis. We often begin the process of putting together a protein with methionine, also known as that first met. So chains of amino acids, they have directionality, okay? The amino end, the N terminus, carboxyl end, C terminus. So you actually do read them in a very, very particular way when they are formed. So let's jump right into the structure of a protein. We know protein structures have 4 basic levels. We begin with understanding what that first main bottom floor structure is, which is the amino acid sequence. This is our primary structure. Okay, so if I want to know what the actual order is, okay, that direct order that's created from the gene, that is the primary structure. Okay, peptide bonds form between them. Okay, and again, remembering it's all about the DNA that determines that sequence and that order. Okay, so there we can see there's a I always pronounce this not right. So it's transthyretin. Okay. Quick thing, look that up. Come in when you do your list. Look up what that protein does. Okay. So secondary structure. Okay, so now we're going to take that initial primary structure, that amino acid sequence, and we're going to begin to twist it and sheet it, and we're going to get regular repeating patterns. We're going to get alpha helices, which are a twist, and then we're going to get your beta sheets. These are all held together via hydrogen bonds between the carbons and the nitrogens, okay? No R groups are involved with this, okay? So kind of keep that in mind. The R groups are kind of out of this piece with the hydrogen bonding, okay? And this is a pretty common thing for the secondary structure. Where the interesting stuff happens, as we head into tertiary, we start to get that very specific shape that's going to form that makes one protein very different from another. Okay, so that chain then begins to take those alpha helices and those beta sheets, and they twist and they fold. And now interactions between our groups, okay, begin to happen. Okay, so here we get back to that transthyretin unit, and you can see that we've got from that tertiary, we now have, or excuse me, from that initial primary, we now have the tertiary. These interactions are also hydrogen bond related. We also get in now disulfide bridges that begin to occur. Okay, and you note them here. So major R group interactions. Okay, again, hydrogen bonding, disulfide bridges form. Okay, and so on. And then some have quaternary structures, specific 3D shapes that are made when more than one polypeptide chain come into contact with each other. It's optional. It doesn't happen all the time. Okay, transthyretin has this as well, 4 identical subunits that link together to create that very unique protein. Okay, so again, we have hydrogen bonding. Again, we have disulfide bridges that are formed. And so I want you guys to think maybe what are some of the reasons behind why proteins consist of more than one polypeptide chain? Why have quaternary structures? What might that do for the actual protein? So there's the quaternary structure. You get those subunits and see how it interacts and kind of creates this kind of funky looking. It's kind of pretty, actually. So functions, what do proteins do? Answer, everything. It's kind of the important answer here. So they're responsible for everything. The organism, life is necessary with proteins. So between the online text, you can go in. Some of these are from the Campbell edition of the biology book. So our text has great stuff in its chapter showcasing protein function and structure. Here's a little bit of what they do. Storage. So you get proteins that are for energy storage, okay, to allow us to have amino acids to build protein down the road. Hormonal proteins. Okay, I would say the majority of our hormones are actually protein in structure. You can talk about insulin is sort of one, that's an easy one that most people are aware of, is one that we have. Structural proteins, this is actually going to create structure. Collagen does this in connective tissue. You got keratin in hairs, horns, feathers, appendages, insects, the silk that spiders weave. These are all structural proteins. Your antibodies, defensive proteins, these are not living things, these are proteins that will actually sit on the surface of the cell in order to combat foreign objects from coming into you. Transport proteins, these are the tunnels and the channels and the pumps and all of that embed themselves into the cell membrane. So very, very, very important to create a transport protein that's in a cell membrane. Hemoglobin is also a transport protein that we've talked about a couple of times already, and that carries oxygen. Receptor proteins. Okay, here's the thing. Not every hormone is a protein, but every receptor for hormone are proteins. So whether you're whatever, doesn't matter what kind of protein you are, okay, or what kind of hormone you are, all receptors that the hormones bind to, or neurotransmitters, or whatever you want to call them, okay, they all act via receptors, and those receptors are proteins. So one can only begin to imagine, just think about it. It just makes absolute sense why we have so many different types of protein. It's amazing. And you have different ones. And then we have contractile motor proteins. Muscle, right? Okay, the actin myosin filaments that are moving back and forth to allow us to look like this and, to do all the stuff that we do and to walk and to move comes from that, as well as the motor undulations of microtubules for cilia and flagella. These are huge, right? And then lastly, not to downplay their role, but enzymes. Enzymes. Do we happen without enzymes? Nope, we do not. So taking a quick look, bringing up sickle cell anemia since we're very familiar with it. This kind of helps. Protein structure and organism. This is that idea of structure dictates function. Change the structure, change the function of the protein. Okay, crazy important is hemoglobin, right? I mean, crazy important, right? It binds to oxygen. Oxygen doesn't make it through our body without it. And it has we've got a couple of subunits here. They all carry the heme group that binds to the oxygen. You can carry 4 per hemoglobin molecule, and that's how it normally works. However, sometimes bad things happen and you get the mutation. It's a single amino acid replacement. You get valine instead of glutamic acid, changing the beta chains, and then you don't have the right structure. Okay? So instead of being a good working molecules that don't ever bounce into each other, they just do their thing, they crystallize and they lose their affinity for oxygen. And you get that well-known sickled shape that occurs. So there it is. You can see the difference between a scanning micrograph of a cell that's normal, okay, and then the cell that's sickled. Okay, they get stuck in the blood cells. The people are extraordinarily anemic. They don't get the energy they need and have, in essence, most will have a shorter lifespan. So we know about proteins, they can denature, right? So they can be broken down. Direct relationship between its conformation, which is its structure, its folding, and the function. If it's altered, you alter the function. Changing the structure denatures the protein. It generally will not work if that's the case. So what can we sometimes do? In very rare, in some cases, not all the time, proteins can renaturate and become and have their shape, but not all the time. So this is something that's kind of important. All right, so when we think about visualizing the proteins, because of their complexity, because of their function, we get proteomics. This is a huge offshoot of like a genomic project. We want to understand how proteins fold. And this is really cool. There is a computer modeling software. This is called Foldit, and it actually helps with the understanding of visualizing frequently used ways to understand how molecules fold. And scientists actually with Foldit got gamers to help build this in order to understand how proteins fold better. So it's actually a really, really neat combination between the scientists and the gamers to develop Foldit to showcase a better understanding of how proteins fold, because it's not this really, there's no real constant rule that exists. But there are other types of protein computer modeling software that's out there, kind of cool apps that you can get that are free to showcase that three-dimensional structure. So, which is honestly pretty cool. So you can see the varying levels. You've got ribbon models, space filling, views of the lysosome protein. I mean, come on, how cool is that, right? So we can get better understanding of how they work based on their shape. And obviously, we can see these buggers with the naked eye. So, I mean, the silk that spiders weave is protein, some of the strongest protein we have in the world. So with that, let's talk about the last group, which are nucleic acids. So general info, storage molecules for biological systems. These are our blueprints made of CHONNP. Kind of recall back to your early biology days when we studied the Hershey-Chase experiment, and that gave us the insight as to the fact that DNA actually is the molecule of heredity and not protein. So there are two kinds, right? DNA and RNA. Nucleic acids have nucleotide polymers or monomers. They are polymers of nucleotides. The nucleotides consist of 3 units: phosphate, sugar, nitrogen base. The sugar is a pentose sugar. It's ribose or deoxyribose. And you have the 4 different bases that exist. So overall, there are 5 bases, 4 for the DNA, 3 of the 4 are the same in RNA, and then we add in the 4th uracil. So here's the biggest differences between them. The sugar is the first one. We have DNA deoxyribose, RNA ribose. The bases, your adenine, thymine, guanine, and cytosine for DNA. And you have adenine, guanine, cytosine, and then uracil and RNA. And whereas DNA is a double helix, RNA is a single helix. So the deoxyribonucleic acid we know serves as our genetic blueprint, okay? All life on Earth uses this as its genetic blueprint. It stores all the information and it is it's heritable. This is what we pass on to the next generation. We know that it is double helix thanks to Watson and Crick's most awesome work that they did to figure this out. They are two chains. One chain runs one way, the other chain runs upside down. They are held together through hydrogen bonds between the bases that make up the rungs of the ladder. The sides of the ladder consist of your alternating sugars and phosphate groups. So you will always find a purine bound to a pyrimidine. Your purines are your double-ringed nitrogen bases, while your pyrimidines are your single-ringed nitrogen bases. I always remember which one's which because thymine and cytosine have a Y in it, and so does the word pyrimidine. There's your tip for the day. So there they are. We're going to explore this much more when we get into our molecular genetics discussions. We all know the story. I think most of you know that I think this is some of the coolest stuff ever, and we know this is probably one of the most important ever discovery that we made, and we think Watson and Crick kind of sealed the deal, but it wasn't without the help of Maurice Wilkins and Rosalind Franklin. Of these four, only James Watson is still with us right now, and at the time of the Nobel Prize in 1962, Maurice Wilkins, Watson, and Crick were awarded. Rosalind Franklin had died in 1959 and therefore could not be awarded the prize, but nobody at this point argues her involvement, thanks to her X-ray crystallography that she did to showcase that DNA was in fact a double helix. So we thank them very much for their contribution. We lost Francis Crick in 2004. Maurice Wilkins just passed away. I believe he passed away in 2000, not too long after Crick. And Rosalind Franklin, we know, passed away several, 1959, quite a while ago due to breast cancer. So thankful to that, we actually have the pattern, that very, very famous X pattern. Yay, photo 51. There's your helix. Gives me goosebumps just to talk about it. It's just, seriously, can you imagine being around during that time when this kind of discoveries and these things were happening? Just the energy just had to be electric. So we'll talk more about that later. All right, so RNA, ribonucleic acid. Okay, RNA is no longer considered just the lackey of DNA. RNA is hugely, hugely, hugely important. And it has a huge amount of, can I say huge one more time? It has an enormous amount of jobs that it performs. We know it acts as the messenger. It translates the information from the DNA out to the ribosome for protein function. It also carries regulatory functions as well as some enzymatic functions. So there's only one kind of DNA, yet we know that there are around about 15 types of known RNAs at current time that you can kind of bust down into about 3 main groups, which is really neat. And so it turns out it's far more interesting than DNA. Once we kind of got our handle down on DNA, I mean, it is what it is. And, kind of coding it was important and understanding the order of the bases. But in terms of what it does one job. I mean, that's what it does. RNA, it's got a whole lot more going on for it. It is less stable. That single helix does make it less stable. It has that one strand, but base pairing can still occur. You can actually get a single strand of RNA that will wrap over itself to create double-stranded RNA. Very important as they look at for things like gene control, turning genes on and off can actually be taken care of through the idea of double-stranded RNA. So that kind of showcases that here a little bit, and we know we can fold it for things like transfer RNA to create the RNA that's responsible for taking the amino acid actually to the ribosome. So we can fold it, it can be manipulated, and it's just downright important. So yeah, can't say enough. But we're going to get into, I'm not going to spend a ton of time here because we're going to be getting to it down the road. All right, so basic information for biology is really important. It is a process. It's a process of matter, the importance of the energy plays, so the releasing of energy, the use of energy, the information that it stores. So DNA stores that information, becomes RNA, so things can happen. This is known as what we call the central dogma of molecular biology, that idea that you have DNA that gets converted into RNA that then creates the protein, right? I mean, this is why we are. I mean, it's because of this. So in its most simplistic nature, that's what it is. Hopefully we can get into some of the more, I don't know, some of the more nitty-gritty stuff about it, and maybe someday this is the stuff, you know, maybe you'll be studying and, you know, bringing to the world information about this maybe someday down the road. So at the end of this all, we want to make sure that you can do these things, okay? Make sure you know this as a combination of the macromolecule vodcasts, that you can do these things, that you understand them, and really don't forget, ask questions when you're here. If there are things you don't get, if there are things you're struggling with, ask. That's why we disco. So, you know, go back, check things out, use your text, look at diagrams. That's the cool thing about these, is that you can see some pretty neat stuff with it. So with that being said, have a great night, and we will see you guys later.